Hello, everybody. This week, I'm going to be going over Lab 9, which is about reversible reactions. The lab itself, I would not say is very complicated, although it is detail oriented, so make sure you're reading the instructions well. The concept of this week's lab is about reversibility and irreversibility in reactions. A reversible reaction, also known as a reaction, an equilibrium reaction, is something where you have reactants A and B reacting to make products C and D. At the same time, your products are reacting to make your reactants. So really what this equation means when you have an arrow going both directions like this is that A and B make C and D, and then C and D make A and B. You guys are probably more used to irreversible reactions because these are simpler and we talk about them more at the beginning of chemistry. These are things like neutralization between strong acids and strong bases, precipitations, and then things that decompose with heat. Once you have done these reactions, they stay as the products. But what is a lot more common is these reversible reactions where your products are also going back forming reactants. So this week we're going to be examining a couple reversible reactions that have distinct color changes. It's a very pretty lab. So in part one, you're going to be working with chromate. So you're going to get 2 milliliters of potassium chromate, and you're going to alternate between adding 3 molar sulfuric acid and 6 molar sodium hydroxide. You're going to notice a color shift as you add drops. And so this is the reaction that we're going to see right up here. It's going to be chromate and hydrogen react to make dichromate and water. And so you'll notice as you add acid, you're shifting your equation this direction. As you add sodium hydroxide, you're shifting it this way. And we'll see a cool color change from that. In part two, we're going to be shifting to work with cobalt chloride. So we start off by getting 4 milliliters of cobalt chloride and divide it between two tubes right here. So each one will have 2 milliliters. One is going to be control so that we remember what color it starts off as. The other one, we're going to add hydrochloric acid dropwise until we notice a color shift. Simple enough. We are then going to get 8 milliliters of cobalt chloride and we're going to add water until we notice a pink color change. And what we're doing over here is this reaction right here. We're adding water to our cobalt chloride and we're adding even more water to the structure, and that causes a change. It makes it pink. We're going to take this 8 milliliters and we're going to split it into 4 different test tubes. 1, 2, 3, 4. These are both the same test tubes. So your first one's going to serve as a control. Your second one, you're going to add hydrochloric acid dropwise until we see a shift to pink. Then we're going to add more water. And once we see another shift, we're then going to add more hydrochloric acid again until we see another shift in color. In our third tube right here, we're going to add sulfuric acid dropwise. And then we'll add water again to see another color shift. And then lastly, we're just going to heat 2 milliliters of our pink cobalt chloride until we notice a color shift. And then we're going to cool it until there's another color shift. So all these steps we're just doing until we notice some sort of color shift. And there's a nice schematic in your lab materials for this week that also help you with this. So I'm going to shift. So now we're going to talk about our questions for the week. So your first data and ask question is asking, why do we use different concentrations for sulfuric acid and sodium hydroxide? So I want y'all to think about why we're using twice the concentration of sodium hydroxide. What could be different in the structure of sulfuric acid that would make it to where we need half as much to see similar effects to our sodium hydroxide? And your second question, you're going to be writing the equation for part two, where it's going to be the same equation that I showed in the slides just a second ago, but you need to place heat in either the reactant side or the product side. So based off your observations, you need to determine which side you need to put the heat on. And then you need to write the full reaction, not just it's on the product side or the reactant side. You need to write the full reaction of heat. So data analysis is pretty simple. And then your discussion and conclusions this week is going to take a while. This is essentially preparing you to write your report next week, since you will have a full lab report next week. So the first thing you have to do is you have to summarize your results, making sure to include all of your data. This says to write 2 to 3 paragraphs. I don't really think that's necessary per se, but what I want to see is I want to see tables of all of your data, And then anything that can't be put in a table, like observations that you have, that needs to be put into a paragraph form. So the main thing is I want to see all of your data in tables, and then I want to see some sort of discussion of your observations, and that would be a good result section. In part two, you're going to analyze your results. This is going to be the start of your discussion. You need to tell me if you think that your results can be trusted and if there are any measurements that don't make sense. So for that, you can compare it to the procedure that you have and the little blurb at the beginning of your procedure talking about the color changes you should have seen. Part three here is that you need to restate your hypothesis. So you should have come up with some sort of hypothesis for what you think is going to occur in this experiment. So let me know what observations either back or reject that hypothesis. Part four is that the main point of discussion is to explain the effect of the independent and dependent variable. So I need you to tell me what is your independent variable and what is your dependent variable, and how does changing your independent variable or affect your dependent variable. Your next question is that in the discussion section, you are supposed to interpret the results that you get. So this wants you to examine your results and try and come up with something on the molecular level that explains them. And so it provides two possible things you can answer here. Why is the acid more effective at changing the color than temperature? And what underlying mechanisms are occurring? So like whenever you're adding water to your cobalt chloride, why does it change colors? What is occurring? So that's what I want you to talk about here. And you are also not limited to these two questions. What I want here is I want you to analyze molecularly what is occurring that's causing all these color changes. In part six, you are supposed to reference something. You need to find a similar experiment and then compare it to that experiment and reference it. If only there was some, I don't know, a manual where this entire experiment has been done and put into writing. Oh no, it'd be crazy if y'all had that on LabFlow or something. In question seven, you are supposed to have a conclusion, which is where you're going to state whether data finally supports or does not support your hypothesis. This should be pretty short and simple. And then lastly, the big question on this lab is that you are supposed to use your answers from questions 1 to 5 to write 2 to 3 paragraphs interpreting your results. In other words, this is your discussion section for this lab report, is what you're doing. So you're really not going to use number one here, because number one was your results section. But question numbers 2 through 5 kind of they kind of are what make up your discussion section. And so what you're going to do here is take your answers from those sections, put them into a paragraph form that flows. I don't want to see just copy paste. It needs to be a cohesive discussion of your results. This is your half of your points for this. So I would definitely try to put a lot of work in here. But that's pretty much your report for the week. As always, feel free to email me if you have any questions. And I hope your lab work goes well this week.